All right, welcome to Principles of Biology 1. I'm Dr. Harries, and today we're going to be obviously talking about Chapter 1. This is an introductory chapter, and I have this split into two videos, two parts. Um, so what are we talking about here in Chapter 1? Well, we've got three main points that I want to cover. So we'll talk about just some background, what is biology, how is life on Earth related. We will talk about kind of some of the big central themes of biology that we're going to encounter through the course of the semester. And then in the second half, I want to talk just a little bit about how biologists study life, and then at the end, talk about some reasons that biology is important for everybody, really. Okay, so this is a biology class. First obvious question then is, what the heck is biology? And as you can see here, biology is simply defined as the study of life. But then that really begs the question, okay, how do we define life? And but before I get into that, I just wanted to remind you, life is generally divided into unicellular and multicellular life. So, as we're going to discuss, living organisms consist of cells. So you have to have at least a single cell to have a living organism. So there are lots and lots of organisms out there that consist of just one single cell that's carrying out all of these functions that are important for life that we will be talking about. But we tend to be more familiar with multicellular organisms because they're larger. We can see them with the naked eye. So most of the organisms that we tend to think about are multicellular. But whether you're talking unicellular or multicellular, there is just an incredible diversity of life on Earth. So almost incomprehensible. Okay, so if biology is the study of life, how do we define a living organism? So as I mentioned already, they are made up of cells. So if you don't have at least one cell, you do not have a living organism. Of course, those cells are made up of chemicals. We'll talk about that in some later chapters. But we do tend to see a common set of chemicals that make up all cells. Um, all living organisms can use molecules from their environment to make new molecules, and they're really talking about cellular metabolism, right? Being able to take in molecules and change them inside the cellular environment in some way. Some of that metabolism has to do with extracting energy from the environment, right? Cells have lots of things they have to be able to do, and those cellular activities require energy, and we have to be able to get that energy from the environment to power that work of the cells. There's always has to be an instruction manual that's basically the genetic information, right, that is going to allow these cells to develop and function properly. So, of course, that is our DNA. And we use that instruction manual, that genetic code, to build proteins. And we'll talk about this in some upcoming chapters, but proteins are really doing all the important work in the cells. So we use the instruction manual to tell the cells which proteins to build, how to build them, when to build them. All living organisms, all cells have to be able to regulate their internal environments. This could entail, how much water is coming in and out of those cells, the temperature, salt concentration, pH, all these kinds of things would have to be regulated to at least some extent. And then lastly, all living organisms exist in populations, and those populations can change and evolve over time. And as we'll see, evolution is one of these central themes of biology. Okay, so we have that laundry list of characteristics of living organisms, and all living organisms get classified into one of these three domains. So these are sort of the broadest umbrellas of life. So three domains are bacteria, archaea, and eukarya. And you may have heard of this distinction between eukaryotes and prokaryotes. That distinction is at the cellular level. We talk about prokaryotic and eukaryotic cells, and we will do that in coming chapters. But I just wanted you to notice that two of the three domains of life are prokaryotic. Again, we are down here, and we're eukaryotes in the domain eukarya. All right. Now, I mentioned there's this just amazing diversity of life on Earth. So how did we get this? And the short answer is we got this diversity through evolution, which, as I already mentioned, is one of these just key central themes of all biology. And no matter what kind of biology you study or are interested in, whether you're a field biologist or somebody who's interested in cell biology or somebody who's interested in medicine, evolution really is arguably the key central theme that really weaves all these different parts of biology together, ties everything together. So we will talk about evolution in more detail towards the ends of the semester, but hopefully you're at least vaguely familiar with the idea, which is that organisms and populations change over time, and sometimes you may hear evolution described as survival of the fittest, right? So it's all about differential survival and passing on your genetic material. And of course, Charles Darwin is credited with a lot of our understanding of how the process of evolution works. And although that definition, survival of the fittest, is okay, even Darwin points out in this quote, he says, well, it's not really just the strongest of the species that survives or the smartest. It's those that are most adaptable to change. And that's really speaking to biological genetic diversity. So species are able to change and adapt to changing environments if they are more diverse. So this diversity is very important for long-term survival, is the bottom line. All right, so we have that central theme of biology. We have this idea that biology is really a big field, right? If it's the study of all living organisms and life is so diverse from unicellular to multicellular, that means there's a lot encompassed under the field of biology. And biologists study life at lots of different levels. So you could study molecular biology, right? What are the molecules inside cells that are important for biology? How does DNA work as a molecule, for example, would be a question that molecular biologists ask. If we arrange these molecules in the correct way, we can form unicellular organisms, living cells, and you could spend an entire biological career just trying to understand how a cell works or some small aspect of how a cell works. If we put cells together correctly, we can organize those into tissues or organs or organ systems and potentially build a multicellular organism. So we could study biology at the organismal level. How does this one particular organism work? Or we could go even bigger. We could think about that organism as part of a population or a larger community or ecosystem. Or if we wanted to go really big picture, we could think about the entire biosphere, which is all of Earth and Earth's atmosphere, basically everywhere on or around Earth that you could potentially find a living organism. So, theoretically, the goal of biology as a field would be to really try to understand the biosphere. So, understand all living organisms on and around Earth and how they tick and how they interact with each other. All right, so if having a cell is one of these key characteristics of life, right? You have to have at least one cell to have a living organism. They're obviously critical to the whole idea of biology, and during this semester, we're going to spend a lot of time talking about what's going on in and around cells. So Principles of Biology 1 tends to focus on some of the micro level details of biology. We'll be talking about, you know, how cells work. We'll be talking about some of the key chemistry that's happening in cells. We'll be talking about DNA and basic genetics. So a lot of what we're going to discuss focuses on things happening at the cellular level. Principles of Biology 2, which is really more botany and zoology, is going to think of things on the bigger level. So since cells are so important to biology and to this course, I wanted to point out that a lot of what we know about cells comes from just looking at them under microscopes. And it was this guy Robert Hooke in the 1600s that really invented the first sort of modern microscope, as we would think of it, and provided some of the very first descriptions of plant cells. You can see some of his original drawings here in A. And of course, you know, microscopic technology has evolved, or the technology of microscopes has evolved over the years, and now we can, you know, take very nice color images. We can look at things moving around inside cells and do all kinds of crazy stuff. But you know, it's that ability just to be able to peer inside cells and see, you know, what's in there and how are these things moving and interacting with each other that gave us that sort of baseline understanding of what cells are and how they work. I mentioned all living organisms have genetic material, right? So, of course, you're hopefully aware DNA is the genetic material. It is that instruction manual. And as I was trying to get across a minute ago, you know, the reason DNA is so important is because it provides the instructions for building proteins inside of cells. And it's those proteins that are really doing all the important work. So we have this, what's called the central dogma of biology, which is this process that happens in cells of going from that instruction manual in the DNA to the actual protein. And it's this multi-step process that includes these phenomena called transcription and translation, where we basically make an RNA copy of the DNA, and then we use that RNA copy to build our amino acid chain that gets folded up into our functional protein. But in later chapters, we're going to go through this central dogma and look at some of these different processes in a little bit more detail. All right, so I wanted to shift now to talking about how biologists actually study biology. And keep in mind, you know, everything you're going to learn during the course of this semester, throughout this class, came from biologists actually doing experiments or making observations, collecting data, and trying to understand some of these phenomena. So even though, you know, it just looks like a fact in your textbook, right, some biologists had to parse out that piece of knowledge. So there's really two main strategies that biologists would use for understanding life. So, and I already mentioned these, but one would be simple observation, and the other would be experimentation. So biology really started as an observational science, right? Basically just walk outside and see what living things are there and start to observe them and watch them and dissect them and try to understand everything we can about them just by simple observation. But really now modern biology is experimental, meaning biologists are designing and running experiments to try to test hypotheses. And that's not to say that modern biology doesn't include observation. It still does. So for example, in this image, you can see this researcher here that is doing a study on bluefin tuna that I'll come back to at the end of this lecture. And they are tagging this tuna with a radio tracker of some type, right? So even though that's using modern technology, it's still observational, right? We're simply going to observe, okay, where does this fish go using this tracker. A lot of biological experimentation these days relies on what are called model systems, and model systems, sometimes also called model organisms. These are organisms that are sort of universally very well studied. They tend to be organisms that are easy to work with in a lab setting. But the idea behind model systems or model organisms is we're going to devote a lot of our resources to understanding this small group of organisms very, very well, right? We basically want to understand them inside and out, even though those particular organisms might not be the most medically or economically significant. So something like, mice or fruit flies would be considered model systems, model organisms. And even though we don't really care a whole lot about fruit flies, as we'll talk about in some later discussions this semester, they're great for doing genetic studies and allow us to understand some key aspects of genetics that we can then apply even to humans, right? So what we learn studying these model systems can be applied to places we may be arguably more interested. So, we could do studies on mice because mice are mammals and their biology is going to, in many cases, be similar to that of humans. So what we learn in mice, we can often apply to human biology or medicine as well. And the reason this kind of strategy works is because all life is related, right? So all living organisms are using that same genetic code. They have cells that are very similar. So it's that relatedness that that again ties back to this idea of evolution that really allows us to study one organism and then apply that knowledge to another. All right, so we can't talk about biologists doing biology without at least briefly bringing up the scientific method. So I know you've probably seen some version of this before, so I don't want to spend a lot of time on it. But the basic gist of the scientific method is that you have some observations, right? These could be observations you or somebody else made, so you have some baseline knowledge. And those observations, that knowledge is leading you to speculate, right? Ask some specific question. And you will usually put that question in the form of a hypothesis, right? And based on that hypothesis, then you could design an experiment to try to test whether or not that hypothesis is correct or not. So you set up your experiment, you collect your data, run the experiment, and probably use some statistical analysis to try to evaluate your results. We'll talk about that in a minute. And then there's really two possibilities at the end of your experiment testing this hypothesis. So either you have some statistically significant results that support your hypothesis, or you don't, right? The results don't support your hypothesis. And you'll notice, regardless of which outcome you get here, you have arrows taking you back up to the beginning, right? So this is the in research. So even if your results supported your hypothesis, you're probably not done, right? It's not like you just solved biology with that one experiment. That's going to lead you to ask some new questions and form some new hypotheses, and then we run through it all again. Or if your results didn't support your hypothesis, then maybe you want to try to redesign the experiment, or maybe you want to change your hypothesis in some way and start over again there. So I guess this kind of is job security for biologists, right? And scientists in general. There's always going to be new questions to ask. And then the last, you'll see down here, very important part of this is that you know, science really proceeds by verification over time, right? So other scientists would need to be able to repeat um, this particular experiment and validate it, basically. Um, so there's obviously lots of different ways you could run experiments, but some of the best, and when I say best, really most definitive experiments tend to be what are called controlled experiments. And so that term can be a little bit confusing because it might sound like, okay, a controlled experiment is going to be one where the researcher controls the outcome, but that's definitely not the case. Really, the idea of a controlled experiment would be one where you're comparing different groups and you're trying to keep everything the same between those groups except for one, what's called the independent variable. So you're basically just changing one variable between these groups. Um, and then you're going to measure the dependent variable. That's basically the response. And the idea is that by just changing one thing at a time, you can really try to get at whether or not that one thing is causing any difference between your groups. And so what I want to do in the second half is start with an example of a nice simple controlled experiment, just to give you an idea of better idea of what I'm talking about here. So we will pick this up in the second video.